Okay, great. Hi, everyone. Thanks for joining us again. Today, allow me to introduce Dr. Taylor Kuhn. So Dr. Taylor Kuhn is an assistant research neuroscientist in the Department of Psychiatry and Biobehavioral Sciences here at UCLA. He is a clinical neuropsychologist and is interested in the relationships between brain structure and brain function across neurodegenerative disorders, neuromedical illnesses, healthy aging, and intellectual giftedness. Dr. Kuhn currently serves as the study coordinator for UCLA's arms of the Lifespan Human Connectome projects. He received his PhD in psychology from the University of Florida and completed his postdoc at UCLA. So please join me in welcoming Dr. Kuhn today.
Thank you so much for having me. So let's screen share real quick. Nope. All right, can everybody see that okay? Awesome. So thank you all for having me. I'm excited to share with you some work I've been doing with Refik Anadol and the Refik Anadol studios here in LA. So we'll be talking about neuroimaging data sculptures, the artistic integration of MRI and artificial intelligence. So we'll go through initially what artificial intelligence and ML machine learning we're using, and then we'll talk about some of the early work that Refik and I have done, including an installation that was just launched at the 2021 Venice Biennale art exhibit last Saturday. And then we'll talk about some ongoing research and art collaborations that he and I have. So if people have questions, you feel free to ask during or at the end. We can make this as conversational or discussional as you'd like. And I'm going to have a bunch of kind of videos to show you as we go through. So this was just a video we put together when we announced the collaboration. And I'll say this is the only time I've ever successfully used social media. So it was through Instagram that we initially connected and created this collaboration. So there's a couple of kinds of machine learning, and basically imagine that, forget about the computer for a second. Imagine you've never seen a car and I ask you to draw a car. So you've never seen one, you don't know what one is, you think, cat sounds similar, so maybe I'll draw a cat. So then imagine you'd only ever seen one car, a 2001 Toyota Tundra. Now I ask you to draw a car. You'd probably succeed at drawing the car that you knew, the Tundra, but you might not be able to imagine the 2021 concept car of the year, right? You'd have a very narrow window of what you'd be able to imagine a car is. Conversely, if I show you a ton of cars, you have a wide training set. These are a whole bunch of examples of what a car looks like. You actually might be able to generate a car, and that's actually a car imagined by a machine that was trained on a large set of images of what cars look like. And that's when you have a single machine trying to predict something. And you can do the same thing with brain data. This was something we did with some UCLA data on HIV. We tried to predict someone's chronological age from MRI-based brain data. And in healthy people, we were able to predict within a year of how old they were. And in people with HIV, where there's this hypothesis that there's an accelerated aging process, it turned out that if you predict their age based on their brain, they look older. All right, so that's a single model. Can you draw a car? Yes. Can you predict someone's age? Yes. But then you can do something called a GAN, or a generative adversarial network. And this is where you take two machines and pit them against each other. So one of them is just like what we talked about. It's trying to predict something. And in this case, it's trying to predict, is this a real picture, like the one I showed you of the truck, or is this a fake picture, like one generated by a machine? And then the one you pit against that is this generator network, which is the machine trying to make an image. So now you've got this setting where every time one machine creates a picture and the other machine says, is it real or is it not? And one of them wins. Either the discriminator figures out this is fake and it wins, or the generator tricks the discriminator into saying it's a real image when it's not, and the generator wins. And by playing back and forth, the generator gets better and better at making an image and tricking the discriminator into thinking it's real. So to use our car example, With 0 iterations, the generator just creates noise and the discriminator wins every time. Every single one of these is definitely not a car. But then as they play this game over and over, the generator starts to win more. And these images usually start to look more and more like cars. And you might imagine that perhaps this one wins and it succeeds in getting a fake picture past the discriminator. And this one, which looks pretty noisy still, the discriminator says, nah, that's fake. And the generator loses. And as it keeps getting better over time, such that most of these look quite like a car. Right? So in this way, you then trained your GAN to be really good at making fake images that trick the discriminator into thinking they're real. And in this way, that's what a lot of the work that Refik has done. So what he does, one of the things his studio does is they take 26 million pictures of national parks and train a GAN that this is what a national park looks like. And so what you're looking at here are all machine generated, or what he calls machine hallucinations of national parks. So this is how he's able to create or train a machine to create art. He can do this. So this is what he called nature dreams, or he's done that with nature. If we go here, he did this with images of New York. And again, all of these are from a machine. None of these are actual pictures. They've all been created by the generator that's trying to beat the discriminator into saying, yep, that's the Statue of Liberty. And you just kind of allow it to continue to do this over and over and over again. And in doing so, you create these beautiful moving pictures. He also did this with images from satellites of Mars, the surface of Mars. And he has a new Mars, the rover and the helicopter that they recently just flew on Mars. He's getting ready to release a new machine hallucination Mars based on that data in the next couple of months. And then what began our collaboration together, I saw him present this, which is machine hallucinations of melting memory. So here they took EEG, so electrical activity of the brain collected when people were in the act of remembering an event, so episodic memory. And they trained a machine to replicate that data, and then they visualized it into what they call a data sculpture. So this is a 3-dimensional moving image of a machine imagining what the electrophysiology of the brain is when a human is remembering something. And it was when I saw this that I reached out to him and said, that's really beautiful. Would you be interested in doing something similar, but instead of with EEG data, with a large set of MRI data? And he got back to me in less than 5 minutes. Not only was he interested, it turns out he's also at LA. He had just recently finished his Master's in Fine Arts at UCLA. And Refik Anadol Studios, or RAS, is in Frogtown. It's east of here. And so we began our work. So one of the things I do here at UCLA is I coordinate the Lifespan Human Connectome project, which is one of the National Institute of Health's largest MRI projects, which means we have a ton of data. We have over 5,000 people's worth of data across the lifespan. So from 0 years old to, I think the oldest person in our study is 104. And that allows us to have structural images, so images of pictures basically of what the structure of the brain looks like, functional images, so data about how different brain networks are engaged during tasks, or how different regions of the brain are connected. And then what I'll be spending a lot of time talking about, which is diffusion imaging. So if you imagine trying to look down on the city of Chicago from a satellite through a cloud, you'd have no idea what the roads look like, right? If you couldn't see the city. But even with that cloud there, if you could see the ways that cars were moving, you'd be able to make a pretty good guess about what the highway systems look like, right? Because cars travel along highways, so you know that that's where the streets are. And that's what diffusion imaging is. We do the same thing, but we look at the way water flows through the pipes or the wires of the brain. And then we color code it by direction. So for example, these big blue stalks are a cortical spinal tract. So that's what goes from the cortex all the way down to the spine and allows you to move your body. These red here, kind of purplish here, that's your corpus callosum connecting between the two hemispheres. right? So we're able to look at the wiring of the brain using diffusion data. And so we collected all of this data from the baby connectome, the developing connectome, the aging connectome, which is kind of a misnomer because it starts at 36 years old, and a couple of these other functional data sets to amass about 5,000 human data sets of multimodal MRI from birth to advanced aging. And we ran them through the basically minimal human connectome project processing pipeline, which I'm not going to talk about a lot, but basically when the data that comes out of the MRI machine, it's real messy. We clean it up, make it really nice and usable data, which includes things like getting rid of skull and getting rid of tissue that's not brain, correcting it for all sorts of distortions. And then we register it all together so that you get these really nice structural maps, these really nice functional maps, and these really nice connectivity maps. Which looks something like this. So here's where we take the brain away, here's where we normalize it and clean up any artifact. We align everybody together so that we know we're talking about the same thing. We make masks of different brain regions. And this allows us to create these beautiful tractograms, which I'll talk about more in a moment, but these are those, the mappings of the highways within the brain. And we use that to both train a GAN to imagine what a human brain looks like, that we could then run tractography on that machine brain and then create three-dimensional realistic forms, both as art and that we could project functional data onto. And I'll walk us through exactly what that means and how we did it. So we took those diffusion images of the brain, which are huge. So the screen you're looking at right now, everything you see is a pixel. If you make a pixel three-dimensional, we call it a voxel. And so these brains, when they come out of the scanner, are large. They're made up of hundreds of thousands of voxels, and you can spin them around and see them in all different directions. And you can also slice them into so you break them down into a bunch of two-dimensional images, which is what you see here. So if you took my brain and then just like sliced it this way and worked your way across, that's what you see. So you get a really small amount and then more and more. Here we're in the center of the brain, and you keep going across until you get out to the other side, right? So you can take it and split a three-dimensional structure into these two-dimensional images. And then we trained the GAN, we trained the machine to make all of these so that we could then recreate a three-dimensional brain. So say again, we trained the GAN just like we trained it to make a car or to make a picture of New York. We trained it to make the map of the brain. So based on those 5,000 images from the Human Connectome project, and then what we got is this. So this is a diffusion map where every voxel has some information about what way the water is flowing, right? So if you, again, just like the car example, if we were looking through the cloud, what way is the highway going? And you can see if you were to follow this track here, you say, okay, the water's flowing there. That's part of our corpus callosum connecting the two hemispheres together. Here we follow this, water is flowing in this direction, so that's another path. This also happens to be the back of the corpus callosum. So we created this image, which is the machine's imagination of what the tractography of a human brain is. And then we ran tractography on that, which allowed us to create a tractogram of this machine imagined brain. And then, as I'll explain in a moment, because of how big that was going to be when we printed it, we also split that down into specific tracks. So like the corpus callosum, which I've been describing. And in doing so, we created a GAN-derived tractogram. So a machine hallucination or imagination of the human brain that looks something like this. And the video you're watching now, after we created this, we sought sponsors for the art exhibits, and one of them was NVIDIA, who makes video graphics and video chips. And in part of sponsoring us, they asked us to present our work at their GTC conference this year. And so we made this for that conference. And so you can see here, and those of you who are neuroimaging experts who do DTI, it's a relatively anatomically plausible brain. There's some things that don't quite make sense, but there's some stuff that is very much very much realistic. For example, right here, that's the corpus callosum that we keep talking about, and to the side is the cortical spinal tract connecting our brains to our spines. As you can also see, the resolution was really, really high. So we were able to create this really pretty but here would be, this doesn't exist in the human brain, right? So there's definitely some artistic expression taken with the machine, but it was able to produce a relatively plausible piece of artwork. So making it digitally was one step. So, but then how are we going to turn this further into art? So what we suggested to the 2021 Venice Biennale, which is both an art and architecture project, was that we would create what we called a sense of space. So we would make this at a scale that people could physically walk through and be inspired by or invited to imagine what it's like to be inside the inner workings of the part of our brain which allows us to have our own inner workings. So the Biennale was kind enough to give us a pretty large exhibit. This is a bird's eye view of one of the, well, you might call them warehouses, but because of where it is in Italy, these are actually kind of antique Roman buildings. And these are all ionic columns. And so what they gave us was a space that's 10 meters long by 6 meters wide and 7 meters tall. And our initial plan was to 3D print something along that scale. Of that image I just showed you. So the machine imagined tractogram of the human brain so that people could walk through it. And we have both here at the Refik studio and also in London where we ended up printing this, are these 3D printing robots. So I think most of us have seen a 3D printed or 3D printer, right? It's kind of like a cube, but it's limited in the size of what it can print. So these robots, which remind me of the robots from Iron Man, one of which in the first movie I like to shoot him with the fire extinguisher, they're like 18 feet tall, and they can 3D print at large scales. So we use that to 3D print this massive structure. And again, we were going to 3D print it and then projection map onto it moving function. So I'll show you that in a second, but we projection maps two things onto it. One, much like that video, is the motion of water through it so that you can see the way that the fibers move. But what we also have is all of this functional data. So data of the way that the brain is active when you remember something, or when you're engaged in a social task, or when you're listening to music that makes you feel something, or when you're feeling a discrete emotion like anger or excitement. And so we process all that data in the exact same way any of us would do for a publication, and we develop maps, functional maps of how the brain is active during those tasks, and we projection map that on to this structure. That way people were not only walking through a static physical structure of the brain, but they were walking through a dynamic, functionally and connectomically alive brain. But then we ran into one small problem, which was that this was going to weigh 8,000 pounds. And in the time that we had to print it, even though we started printing in February and May 22nd was the opening date, we weren't going to have time to get the Venice government to approve the weight and ensure that it was safe for people to walk under. So we ended up having to pivot a little bit for this particular task. Thankfully, Siemens, who makes the MRI machines we use, is interested in having us print a slightly smaller version of that for their their Munich headquarters. So we'll still get to print the full scale tractogram. But for the Biennale, we had to make some changes. So if you take that initial image and start to remove some degrees of freedom, remove some complexity, you can backscale it to just specific tracks, right? So for example, here's that cortical spinal tract I keep referencing. Cortex here, sensory and somatic cortex there, down here is where it joins with the spine. Right here, that's the decussation of the pons, which is where it crosses. So we all know the left side of our brain controls the right side of our body, and it's literally because of these fibers cross to the other side right there, right? So we were able to take our full-scale tractogram, segment it down into specific tracks, and then 3D print those tracks so that we then had smaller, more manageable pieces, which we planned to install a slightly different way. So this is an example of those 3D printing robots. This is a recycled polymer. So all of these materials we used were recycled. They were then melted and then printed using these arms into these forms. And each of these that you see on the right are components of each of those individual tracks. So again, when we print the full scale tractogram, we'll be printing it in kind of chunky segments that will be put together into a single unified form. But for this, because we couldn't have an 8,000-pound structure sitting on the ground for people to walk through, what we ended up doing was printing these different tracks. Now, I don't entirely understand why they thought this would be safer, but what the Venice government was comfortable with was us stringing all of those tracks up from the ceiling and then projection mapping onto that. So this is it in Venice, Italy during the installation. This is Refik here, and this is some of his team. And this is as they're working to get each of the pieces up and strung in correct place. And before I show you the finished product, I mentioned projection mapping. So I wanted to give you an example of this. So here's a static rendering without color. For those of you a little less familiar with brain anatomy, so this is the brain seen from the side, like this. So the eyes are here, looking that way, looking this way. And again, if we had just 3D printed the structure, it would look something like this, but we wanted to give it depth and dimensionality as well. And as I mentioned, one of the ways we did that was by showing the movement, projection mapping onto it, which for people who aren't familiar, it's exactly what it sounds like. You take a bunch of projectors and you map what the structure looks like to the projectors using some software, and then the projectors project information onto only those structures. Refik has a really beautiful TED Talk, and in that TED Talk, he shows what was his master's thesis. Some of you may have actually seen it in 2018. He projection mapped onto all of the Disney Concert Hall in downtown LA, which for those of you who've seen it, it was designed by Frank Gehry, who was this incredible visionary architect. So it was not as simple as projecting onto 4 or 5 sides of a rectangle or a cube. I think there were 46 different sides of that building, which required like 120 projectors to projection map onto this building. So that's the kind of technology and the kind of process we're talking about. And in order to do that, we created these latent flow, which is basically to say we created videos of how water moves through those different pipes. So you can kind of see as you move from top to bottom and front to back, these videos bring life to those structures. So to play that one again, where you see coming into the central layers, you're going to see that cortical spinal tract. And then you see the corpus callosum. There's our strong cortical spinal tract, which I keep using as a landmark. And it's moving from front to back through the brain to really bring the sculpture to life. We did this when we created multiple latent flows. So this is another example, but coming from side to side to really, again, bring this to life in an anatomically plausible, data-derived, empirical way. We really wanted to maintain as much realism in this as we could. So there's that side image of the brain we've seen before. And that's what it looked like when we finished. So that's it hanging and with projection mapping onto it. So there will be because we are currently wherever we are in this global pandemic, the Venice Biennale is not open to the public, sadly. But they will be live streaming the exhibits in the next several weeks. So for that reason, I was asked to not show video of it yet. But I'll send out the link as soon as it is live streamed so that we can all see it. But that's it being projection mapped onto. And as you can see from their height, Refik is probably 5'10". So you can get a scale of how high this space is. This is, I think, about 25 feet to the ceiling here. You see those beautiful ionic columns I was describing. And so a couple more action shots. So this was our Venice Biennale piece. And one of the things that we were really excited about was in going through this piece, we really created a foundation of an ongoing collaboration. They now have all of these neuroimaging pipelines and tools that we worked on together to build. So we went through working with Siemens and NVIDIA to get funding. So we have really this amazing foundation to do a whole bunch more neat things now. So from an artistic perspective, we're going to do some physical data sculptures and some digital data sculptures. I'll show you some of those in a second. We're going to work with a dear friend and Tai Chi brother of mine is the artist in residence at the Internet Archive. So we're going to do a collaboration with them. And then we're working on a real-time EEG infinity room, which I'll describe in a moment. And we're also working on a myriad of publications together. So physical data sculptures are a lot like what I've just shown you. So we're going to 3D print something and then projection map data onto it. So for example, this and this are both examples of a physical three-dimensional data sculpture based much like the melting memories piece that I showed you from EEG was on a 3D rendering of the EEG or MRI data, and then we'll projection map data onto it. Here we've modeled the functional networks of an in-back task or a working memory task. Here we've modeled a social cognition task. You'll see me show you those again in a moment. So we're going to continue to build physical models that we represent and map functional data onto. Towards this end, it's been really fun kind of reaching out through the scientific community and asking for donations of data. So we now have data of humans experiencing, I think, 8 or 10 different discrete emotions. Dr. Builder here at UCLA has been kind enough to share the Big C project data with us, which is a really cool project on exceptionally creative individuals, which includes visual artists and musicians. So we have data collected in the MRI machine of people doing things. For example, they took some famous musicians, put them in the scanner, and then asked them to imagine they were playing a song that was automatic. So acknowledging that this is a little culturally insensitive, something like Twinkle, Twinkle, Little Star, right? Something that people from certain backgrounds have heard so many times it's become automatic the way the alphabet becomes automatic. And we compared that to, we then asked them, okay, now imagine playing a jazz solo you've never played before. Impromptu, give us all you've got as an exceptional musician. So we have functional MRI data on people being exceptionally musically creative. We also have MRI data on people listening to music that makes them feel emotions. And we can contrast that to listening to sounds that don't elicit emotions. So, and we're also, one of the other things where the one thing I don't have yet is data on people dreaming. So we're still seeking out data on people dreaming. And I think we have some data that is getting ready to be shared with us on people experiencing hallucinogenic states. So we thought if Refik is calling these machine hallucinations, it might be interesting to have the machine hallucinate using data from humans hallucinating. So one of the things that's really exciting about that is we now have all of these different discrete emotional and cognitive and creative tasks that we have imaging data on that we'll be able to integrate into our art. And in that same way, we can do this digitally. So we can make images, much like the New York and the nature and the Mars images we showed you, where they're two-dimensional. We can also create these rich three-dimensional images, like the initial image I showed you of the tractogram that very much came to life. So our ability to create digital data sculptures is really robust. And I'm going to circle back to this in a second when we circle back to some of the research that we're going to do together. So the Internet Archive is a really, really neat place. It's up in San Francisco, and it was founded by the person who owns the patent for Amazon's Alexa. So his name is Brewster. As you imagine, that was a very lucrative thing. And so he's been able to do a lot of things that he's really passionate about. And one of them is the Internet Archive, which has the mission of archiving the entire internet, but it has the broader mission of archiving everything. So for example, they back up the public facing internet like 260 times a day. And that gets used often, including in politics. So for example, if a politician makes a statement, this was true very much in the last 5 years, but we don't need to get into that, but if a politician makes a statement about, oh, Google didn't do this thing, or somebody said something on the internet, if the Internet Archive is able to go back to exactly that date and time and show it. If you go to the Internet Archive, they have this really neat exhibit. It's 6 cathode ray tubes, like the old TVs with the curved fronts and the gigantic backs. They have 6 of those. It takes about 4 feet. And each of them is cycling through the entire internet from 1996, and which are hosted on like 8 hard drives that are no taller than the TVs. And it's really interesting to see that. But so they archive the entire public facing internet, but they also are trying to archive everything. So they archive books, they archive movies, they archive personal things that they're able to get. So for example, they're working on a project where they have this massive set of home movies from this indigenous community in Central California from like the 1930s to the 1970s. And so they're archiving that, and then artists such as Amir work to put together a meaningful art project from that to share with the world and help kind of bring this data to life for people. So Amir is a tenured art professor up in San Francisco. He's also the director of special art projects at the Internet Archive and their artists in residence. And he and Refik and I have met a few times. And as I'm sure has been clear, whether it's 26 million pictures of nature or 11 million pictures of the surface of Mars or 5 million data sets, 5,000 data sets of the human brain, Refik's art is really based on large scale data. And so the Internet Archive is arguably the largest scale data set in the world. And so our ability to do massive data visualization projects and art projects together will be pretty endless. So we're very excited about that. And then lastly is this real-time EEG infinity room. So EEG, right, electrode cap, able to collect real-time data about the electrophysiology of the brain. It's been around for a long time. It's relatively robust in terms of, you can get reproducible findings. If you show somebody a certain task or show them a certain image, you can get reproducible findings. And since about the 70s, it's been used in artwork. So one of the first places that this happened was EEG-based music. So this was, I think, inspired by, CERN, the Hadron Collider, makes most of their data public. And that includes from when they crash particles together. And a mathematician in, I believe, the late 70s created what he called the sonication algorithm. Or sonification algorithm. And so what he did was he took the data of 2 particles crashing together, and he created an algorithm that turned that into sound. And then they did a little mastering and created music from it. And so in a very similar way, people have created sonification algorithms to turn EEG data into music. And one of the things that's really neat about that is if you do that in real time, then imagine you're the person who's wearing the EEG cap. That EEG data is being collected in real time, converted very quickly into music that you then get to hear. Well, what happens when you hear it? Well, when you hear the music, you go, oh, wow, that's so weird, or how neat, or how beautiful. Whatever reaction you have changes your brain state, which changes the music. And so it becomes this really neat feedback cycle. And they've actually done some exhibits or art installations where they've had groups of people wearing EEG caps and listening to the music. So you become an orchestra creating EEG music together, which you can imagine somebody laughs and somebody sneezes, and you have all of these different changes to the music, which just contribute to more brain changes and so on. And so in that same way, we want to do a similar project. So Refik has, I think it's like an 11 by 11 foot cube. It's an infinity room. It's been in 48 states in several countries. And it has projectors in it that allow them, it's not, I think a couple sides are mirrors, but it's mostly projectors that project onto all 6 surfaces that give it the infinity room experience. So not quite the entirely mirror infinity room that the Broad Museum has, but a similar effect created by projectors. And what we're going to do is have the people inside the room wear the EEG cap, have their EEG data decoded in real time and converted through these same kind of GAN algorithms that we've described into moving images, into data sculptures, and then projected into the inside of that infinity room so that a person will be immersed in all directions by art generated from their brain in real time. And then just like the music, when they see the art, it'll change their brain. When their brain changes, it changes the art, and it'll become this really fun and recursive art installation that we may add the layer of EEG-based music to as well. So we're really excited for that project. And then just real briefly, I just wanted to mention some of the research we're doing. So we also want to publish some of this. So we're going to publish some of the GANs that we use to create this 3D tractogram and try to publish on that 3D tractogram. And then because we have this ability, so I've never thought I'd get to be involved in art, and I'm having a great time and incredibly humbled and honored to have this experience. And as a scientist, which is the core of what I do professionally, how could I not try to capitalize on the access that we currently have to these visualization abilities? I think one of our hurdles as scientists is to have as meaningful an impact as we can. I've written some papers that I'm proud of, but I don't know how much impact they've had on the lives of people, and I'm pretty sure it's very, very small. It's part of why I maintain a clinical practice, because I know I'm having a meaningful impact there. And one of the ways that I think we can have a more meaningful impact with our research is by making it as easily digestible as possible. And one of the best ways to communicate information is visually. So we're working together to do some what we hope will be both very elegant and simple, but also profound visualization of data. And I'll give you an example of that in a moment. And then we're also looking at the possible utility of some of his artwork. What happens if you look at that art on brain function, and it's possible to use that as a wellness tool. So if we switch back to thinking like scientists for a moment, we have lifespan MRI data. And part of what we've been looking at is what happens across the lifespan. So the Human Connectome project is all healthy adults. The data, there's like 12 pages of exclusion criteria. So everybody who's recruited into the project, meets a very narrow criteria of healthy. And so we have this data set that really allows us to look at the typical or the typical development of the brain and the typical maturation and aging of the brain. And papers have been published that look kind of like this, that show you, okay, at 8 or 11 years old, the structure looks like this. You can kind of look over here and see, that looks a little bit different as we get older, looking a little different as we get older, but it's not super easily digestible. So what we're going to do is use the visualization tools that we have at our disposal through our collaboration with Refik Anadol Studios and turn this into a data-driven movie where you can watch the brain from zero begin to develop, begin to mature, and then begin to age. And then and this isn't a GAN, this is the actual ground truth data. And then we'll be able to do things like color code regions of the brain based on what's driving it. So if you imagine watching this small brain get bigger, develop more grooves, get bigger, mature, and then start to atrophy, that's really interesting. But if we then layer with, imagine it's 5 different colors, and yellow is the things, the changes that are driven by genetics, and blue are the changes that are driven by education, and green are the things that are driven by psychosocial factors like parenthood, right? We can really start to create a visual narrative that is digestible by basically everyone and tells a really, really important story about the growth and maturation and degeneration of the human brain. We can do the same thing functionally. So the MBAT task, as I mentioned earlier, is a working memory task. And you can see how the functional network of the brain that's required to do this, to perform this task, changes across these different epochs of life. But again, if we could watch that as a movie and see how that evolves, how the network moves. I remember learning, as a concrete example, I remember learning that as infants, we process language bilaterally, and then the right hemisphere activity that's involved in processing language, some of it, I was taught when I was in college that it was all of it, but we know now it's only some of it migrates over to the left side. And so to be able to watch a ground truth data-driven video of that migration happen, of the change in that functional network across the lifespan, I think would be really profound. Same thing is true, we could do that with any network. So this is another example, is the shifting in social cognition networks over time. So I think that there's a scientifically powerful opportunity with this collaboration to show our empirical results in a way that hopefully has a higher impact. And then lastly, so we can look at structural connectivity a little differently. So this one over here looks the most like a brain. The eyes are kind of here looking up. And here what you've done is you've used something called graph theory to figure out what the different structural networks in the brain are and then color code them. You may have seen maps like this, either of like your social network on Facebook and how you're connected to different groups, or of airports. If you look at like a map of the US, you can see what airports are the hubs. There's a ton of planes coming in and out of LA because it's a Delta hub, and a ton coming in and out of St. Louis because it's a Southwest hub, and other airports have smaller regions. So you can plot and visualize an entire network, whether it's an air system or a brain, using these visualizations. Some of the work I do is in neuromodulation, so trying to shift how the brain is active using external energy like ultrasound. And so to be able to use these kind of visualizations to show, okay, this is the brain connected at rest. This is how we've shifted the brain's connections following applying energy like ultrasound. I think it'll really improve our ability to communicate our findings. So that's basically it. Special thanks to Refik and his studio. A special mention to Rishab, Arda, and Nick, who were the three people I worked with most closely to bring the Biennale and the GTC art to life. Also, a huge thanks to the Connectome project, including the UCLA PIs, Susan Bookheimer, Roger Woods, Mirella DePreto, and Gary Small. And then the work in the Refik Anadol studio is supported by a lot of sponsors, including, as we've mentioned, NVIDIA and Siemens, UCLA, Harvard, Google, DeepMind, etc. So thank you all for your time. And it's been a lot of fun to get to show this to you.
Thank you so much, Taylor. That was absolutely fascinating. At this point, we can open it up to questions.
So when you generated the flow through the structure, was that adversarially generated as well? Or is that just the structure generated and then the flow was computed?
Good question. So the structure was generated using the GAN, and then we just flowed through that. We mapped basically the fiber direction, and then just flowed along the fiber directions from the diffusion map we got from the GAN.
Cool.
I have a question. Oh, go ahead.
Absolutely. Sorry, just one sec. But following up on that great question from Kyle, the functional activity that we projection mapped onto it. So, for example, what parts of the brain were engaged during a social network task or during the working memory task? Those networks were generatively created by a GAN. So we trained the GAN on 5,000 people's data set of what's the brain network that's involved when you're socially active, or what's the brain network involved during a working memory task. And then the GAN created its own functional network. And that's what we projection mapped onto the structure. So it was a machine imagined structural connectivity, a machine imagined functional connectivity. But then within that, we followed the pathways that came from that. Yes, there was another question.
Yes. First of all, thank you so much for this inspiring talk. And I was wondering, I'm not sure how much time it takes you, like weekly or like the entire collaboration took you for the project, but I was wondering what your initial motivation was or is still to initiate this collaboration. Were you always interested in the intersection of art and science, or did the opportunity just present itself?
Thank you. So Refik and I started working together in about November. I'm a drummer, but not an artist, but I do really love art. And I had once before done an art project. There's a local, so that the example I gave of the sonification algorithm from the Hadron Collider, I learned about that from a local artist I met at the Brentwood Art Fair, back when they used to, once a year they close San Vicente down and have a bunch of artists. His name's Michael Bermudez, and he's a painter who does work that's based on math, music, science, and chaos. And he had done a piece called the sonification algorithm based on he listened to the music that was created by the sonification algorithm from the Hadron Collider's data and was inspired by it and painted it. And I just thought it was so beautiful. And he's himself an inspiring, fascinating artist. And when I transitioned from postdoc to faculty, I asked him if he would do a painting with me. And so as many of you who may be grad students or work in MRI labs know, we get scanned a lot, right? Somebody needs to set up a new project, and so you hop in the scanner to help them out. So I have a lot of MRI data of my brain, and I ran tractography on my brain, and he and I worked together to create a painting of the tractography of my brain, and he had this amazing idea to pick regions of the brain and then put things next to them. So between my Broca's and Wernicke's area, so between the part of my brain that comprehends information, or verbal information, the part of my brain that allows me to produce speech, we put my favorite poem. Near the part of my brain involved in music, we put my favorite guitar tab from an Incubus song. Right, so Michael Bermudez and I worked together, though all artistic credit to him, to create a painting that is now in my office that I really love. So I dabbled in that way. But really, I was just fascinated by Refik's work. I'm not much of a social media person, though I love to lurk on Reddit, but I don't really, engage that way. But I had found his artwork and I had been following it. And I just think it's really inspiring and really beautiful. And when I saw Melting Memories, I guess I was just a little impulsive and I reached out. I didn't really expect it to work out, but it like I said, he got back to me really quickly, and he's so enthusiastic and so inspiring, and really, he's probably installed 6 art installations around the world since in the brief time that I've known him, including Melbourne, Australia. I believe he was in Copenhagen, here in LA at the NASA's Jet Propulsion Laboratory, the Venice Biennale, and then he's also working on with the distillery in Scotland, I think, right now. So he's just massively prolific, and so I think as much as I was excited to get to do this work with him, so much credit goes to Refik for his kind and inspiring personality and his kind of unstoppable creative genius.
Hi. Yeah, thank you for coming. That was really exciting. I really resonated with what you were saying about being able to translate the excitement of these findings to a broader audience. And it's easy to appreciate how things like fMRI data, which is inherently already very beautiful, that sort of transition into the public art sphere. I don't want to use the word easy, but there's a more direct maybe translation there. And so I work a lot with gene regulation data, like Hi-C, chromatin conformation capture. And then so, sometimes from that you get resulting gene regulatory networks, which are similar to the networks that you were showing. But I'm wondering, it's really appealing, the idea of being able to, make this aesthetically pleasing and show to an audience to have these concepts, biological concepts that are interesting. So I was wondering if you had any thoughts on how to approach that for things that were maybe more difficult to translate into the art world.
Yeah, that's an excellent question. I'll think about it more, but kind of spitballing, I always look at labs that have been around for a long time and are really well funded, and just imagine that they have a technical writer and or graphic designer on staff, and how amazing that would be. And so part of what's been exciting is to have access to these people who are so great at visualization and who are also so inspired or interested in neuroscience. So I feel like collaborations can be really helpful in that way, I guess, is part of what I'm saying, is finding people who have that skill set, even if they themselves aren't necessarily scientists, but they have that skill set and are interested in collaborating. That's one thing that comes to mind. I think that that there's also a big push. So in the same way that there's a big push for visualization, there's a big push for reproducibility in our field right now. And I think that getting a little bit creative about the way to present data, both in a visually appealing way and in a reproducible way, is really nice. So to the extent that we can create and share visualization tools, Refik has been really generous in suggesting that we could create software for visualizations and then share it. So I'm hoping that we are able to do that kind of thing and make these tools. You know, in the neuroimaging world, like we all sit down and we use like FSL or FreeSurfer or AFNI, and we all write our own code to do the same thing that 80 other labs are doing. So it's really nice when we can share code that we've already written, so we don't have to start from scratch. And so perhaps there's a way that that we can, as a community, get better at making things like that public. I don't feel like I'm giving you an amazing answer, but I will keep thinking about it.
No, I appreciate that. And yeah, I guess this is just a thought, but I think the idea of like, how do we translate to this a wider audience? Like even within UCLA, in my academic department, if people aren't specifically working with gene regulation data, a lot of times, like I know that people will see something like Hi-C and doesn't translate at all. So there's even a barrier within people who are studying similar things where sometimes the data representation is super unintuitive. So I wonder if, yeah, I guess this just gets me thinking about even within the scientific community, if it'd be nice to be able to share our data in a more intuitive way.
I completely agree. I mean, if for those of us lucky enough to try to publish somewhere like Science, we have to communicate to the broad scientific community. And I've seen people try to do that in over elegant ways. Like you get these, you know, a 3-dimensional map. 2D bar graph would have been plenty, but now it's a 3-dimensional wave graph. And I'm like, what am I looking at here? So I think trying to think about how to communicate it simply is really nice. So that image I showed, that was the graph theory. So that's 3 dimensions, and it's 3-dimensional, and it's color coded, and you can make the each edge, each little square that is connected by the lines, different sizes. So you could communicate so much information by saying, all right, X, Y, Z, the 3 dimensions allows me to create a structure that looks like a brain. And then I can represent whether it's Atlanta and LA, you know, a big airport, a big hub, or I don't know, Des Moines, like a little one, simply by the size of my cubes. And I can then color code them into 3 or 4 different networks. So in that way, you're able to communicate a lot of information relatively simply and visually. And I've seen Refik Studio do that with, they represented some data from a library, and they were showing like all of these different, kind of like a word scatter plot in 3 dimensions and 3 colors with different sizes. So you're able to try to make something really simple, but I don't do genetics work. So I'm not familiar with Hi-C, for example. And to try to communicate that, to me, it would have to be relatively simple and straightforward, or I would have a ton of questions, half of which wouldn't make any sense to someone who knew the field. So I think, you know, I was wrestling to explain to my lab something recently, something relatively simple about recruitment. And I finally thought about it in a way that I could communicate it well. And part of what I think was helpful was shifting from thinking, okay, I get this and I'm communicating in a way that makes sense to me, to thinking, to realizing that I didn't matter in the equation. Some of my staff were having trouble understanding it. So how could I communicate it in a way that landed for them? So really shifting from being focused on myself as the communicator to being focused on them as the people I wanted to communicate to. That shift allowed me to come up with a way to communicate to them in a way that actually worked. So that's one other thing that comes to mind.
Thank you.
Thank you again so much, Taylor. I have a question, since a lot of us here are like grad students and early career researchers who are interested in exploring like paths that incorporate communication in addition to academic activities. I'm very curious about how you balance these artistic projects with your responsibilities as a clinician and a researcher. So yeah, I guess, what does your day-to-day look like, and like the time commitment to this project, and like how do you balance those things?
Excellent question. So delegation, a lot of this is delegation, and I try to do good task management and prioritization. So like seeing patients and attending to my UCLA research responsibilities, that comes first. I have 4 or 5 Slacks that I use for project management. So like we have a Connectome project Slack. We have this Refik Anadol Connectome project Arch Slack. So I use project management tools like Slack, which are really, really helpful. But as I held on to being the person to like do the stats and do the research as long as I could. And I still have my toe in it, but at a certain point you end up shifting into kind of a lab management or project management position and writing more grants and overseeing. So, as coordinator for the Connectome projects, I'm rarely the one, running a neuropsych battery or sitting at the console of the MRI scanner. I'm much more so overseeing 7 staff who do that. So there's a lot of it is about successful and effective delegation and communication and project management through, I use Slack. Other people like Trello. Personally, I don't, but whatever the system, it doesn't matter what I like, it's whatever the system that works for you well is. And then, this is it's a passion project, so it takes the back burner. But I end up having a couple meetings a month. We're in a bit of a lull now. There was a big push up into the mid-January to get the GAN tractogram so we could send it to the 3D printer in London. And then there was a big push kind of between February and March to get the GAN functional networks so we could put the projection mapping together. So there'd be these little pushes that were kind of intense, and then there'd be a lull. But for the most part, I was predominantly, I collected data and I oversaw the transfer of the data, and then I oversaw and directed the people who were actually running the imaging pipelines and running the GAN, and so it was really almost like a consulting role.
That makes sense. Thank you.
Yeah.
And we are coming on to the hour at this point, but I think we have time for one more brief question.
I'll ask a fast one. Taylor, that was so awesome. Thank you so much. I guess, is there anything that you have learned or realized through all these projects that were surprising to you, or that like some sort of insight into like the actual data or the brain in any way that you thought was unexpected through these new visualization tools?
Cool question. Thank you, Zoe. That's a good question. Well, I didn't know. I've published in some machine learning stuff before, like that brain aging prediction I showed, but I had no idea what a GAN was. I had never. So I've learned a lot about machine learning. In terms of the actual imaging data, not yet. But when we finish putting together those movies showing how the brain changes structurally and functionally across the lifespan, and color coding it based on our predictive factors, I'm sure I'm going to learn a ton from that. I think there's been a lot published on, we know cortical thickness increases. We know sulcal formation and invagination increases over time. And that kind of the reverse happens. It's age-related or neurodegenerative related demyelinating processes occur in things like Alzheimer's disease. So I think we kind of have these ideas fundamentally based on some longitudinal data that we've seen, we all have read, but I haven't seen it. I haven't been able to really engage with it the way that we'll be able to soon. And so I expect to be really inspired by seeing that. And I think the same thing will be true. I'm very excited to use these tools to show, like my ultrasound work, for example, we know we're having some exciting success modulating brain perfusion and functional connectivity with some downstream behavioral and psychophysiological effects, but to really be able to visualize these networks changing and how that is driving these behavioral and psychophysiological effects, I think will be exciting. So great question, not yet, but I think soon. I know it's 11 o'clock, everybody. Thank you so much for having me. It was a real pleasure.
Thank you so much for coming.
Thanks, Taylor.
Yeah, thanks again, Taylor. And a reminder to everyone in attendance, we will be